Well, hello and welcome. So glad to have you here with us. Thank you for taking the time out of your day to be here. We've got a great topic to cover. We're really excited to chat with everybody about fluorescent imaging and how MVF software tools can help you acquire powerful images for your research. To get started, my name is Nathan Leese. I'm the product manager for most of our acquisition products. So Neurolucida, Stereo Investigator, Microlucida. I've been here with MVF for about 19 years. I'll start off with some introductions of our topic, our format, and our presenters, and then so info on how to submit a question and interact with us during the webinar. We're going to start off with some background and theory behind fluorescent imaging. Then we'll get into practical use within our software. We'll finish with a Q&A session. Today, we have Jean Fecteau, a Biosystems specialist. He's a senior member of our on-site training and technical services team. He's been with us for almost 7 years. I'm sure some of you have called in and talked with him over the years. And we also have Jordan Deso. He's a software and hardware support engineer. He's a newer member of our team with a software background and specializing in our hardware solutions. He's been with us for about 2.5 years. Next, I wanted to let you know how to submit a question. You know, after using Zoom for the last 3 years, as much as we have, I'm sure everybody knows how to do this. We just want to cover it. Make sure you know how to do it. So bring your mouse down towards the bottom of the screen and a little interface should pop up. Click on the Q&A button to open the question and answer panel. Type the question and press enter. You're welcome to submit a question throughout the talk as you think of it. We'll try to get to as many of those questions as we can. If we don't get back to you during the webinar, we'll try to address those questions afterwards. Now that I have all that out of the way, Jean, I'll let you take it from here. Give us some knowledge.
All right, thanks, Nate. So first, before we start taking a deep dive, I want to just start with some baseline knowledge on what we mean by fluorescence. Typically, we're talking about a particular particle, a fluorescent particle, that when excited by a particular wavelength of light, it will then emit a different wavelength of light. And to get a little bit more specific, the particle is always going to emit light that has a lower energy than the light that excited it. And since a photon's energy is inversely proportional to its wavelength, we should expect that the excitation light will have a shorter wavelength and the emitted light will have a longer wavelength. And that translates to the excitation light being closer to the blue side of the spectrum and the emission light being closer to the redder side of the spectrum. And just to give a little bit of interesting background on fluorescence, this has been sort of an ongoing project for the past 60 years between many different bioscience fields. It started with discovering and isolating the green fluorescent protein, or GFP, in 1962, which I'm sure many of you have probably heard of before. And then in 1999, we discovered and isolated DS red, which is another fluorescent protein. And what I want to point out here is that in 2008, all of this work resulted in a 3-way split of the Nobel Prize in Chemistry. So this has had a lot of impact on many different fields across many different biosciences. And some of the eagle-eyed among you may have already noticed that we don't just have the green and the red. We've also found ways to modify these proteins so that they can emit different colors. And so we really do have now a rainbow across the visible spectrum of different fluorescent particles that we can use. To look a little bit more closely at one in particular, looking at the GFP spectra, well, I've mentioned that with fluorescence, we're typically looking at a particle that is excited by a specific wavelength of light and then emits a specific wavelength of light. But the proteins and the molecules that we tend to use in fluorescence are a lot bigger and more complicated than a simple particle. And so these excitation and emission wavelengths tend to actually occur as more of a spectra with a particular peak where the protein is most responsive. So with GFP, the peak for the excitation is around 475 nanometers, which is a bluish color. And then the peak for emission is around 510 nanometers, which is a greenish color. But as you can see, those spectra for excitation and emission have a very wide range of wavelengths that we can also expect to get. So it isn't just going to be as clean as a simple one wavelength in and one wavelength out. And just to pause here for a bit of nomenclature. Up until this point, I've been referring to the whole protein as a fluorescent protein. But specifically, the part that actually does the fluorescing is a small molecule that's contained within the middle of this protein, and the rest of the protein is sort of encasing and protecting this fluorescent molecule. Now, the molecule that actually does the fluorescing is something that we'll refer to interchangeably as a fluorochrome or a fluorophore, and this is terminology that you're going to hear me and Jordan use throughout the rest of the webinar. So let's just talk a little bit about how fluorescence tends to be used in the bioscience field. Typically, one of the techniques that we can see is that a specific molecule is identified that is a fluorescent molecule, but also happens to bind to an interesting biological target. There aren't a lot of different molecules that we use in this particular category, but the ones that we do tend to use tend to see a lot of very common usage across many different fields. Another technique that we use is called immunofluorescence, where we employ the use of antibodies to target more specifically something that is of biological interest to us. And then the antibodies will also be conjugated to some fluorescent molecule or protein that will carry the fluorescence and be used as labeling. And there are two different methods we can do, where direct immunofluorescence uses just the same one antibody for both the targeting of our biological target and also to be used to carry the fluorescence. Whereas with indirect immunofluorescence, we actually use two different antibodies, where the primary antibody will be targeting and binding to our specific target, and the secondary antibody will actually do the carrying of the fluorescent molecule. We can also see transgenic subjects used sometimes, where the genome of the subject itself has been altered such that expression of our target gene of interest is also going to result in expression of a fluorescent protein. And then finally, we can also sometimes see gene transfer using viral vectors, and these can get very experiment dependent in terms of what they want to do. Now let's look at a couple of these a little bit more in particular and some of the more common methods. One of the molecules that we tend to use both to bind to targets and to emit fluorescence is DAPI, and this stains the DNA in nuclei, which makes it not only great for visualizing neuroanatomical boundaries of the brain, but it also makes it very good for counting cells. So for these reasons, it's also typically used in conjunction with a lot of other fluorescent techniques. With indirect immunofluorescence, we can take advantage of the primary antibody by finding a very specific binding target, taking advantage of high binding specificity of antibodies. And we can use our secondary antibody to carry basically whatever fluorescent molecule we're interested in. And this allows us to build up a whole bunch of different antibodies on the same sample, creating a rainbow of fluorescence on the sample itself, where each color is going to give us some different information about the biological effect that we're investigating. And then finally, another technique that we tend to see fairly often is the use of a rabies viral vector. And this can be implied as an injection in vivo, where the effect actually becomes localized. And so this makes it really good for retrograde labeling and tracing projections of neurons that project to the injection site. Now, this is by no means an exhaustive list of techniques. There's a lot that I haven't spoken about here, but ultimately it's up to you as to which technique you want to use. As long as you're able to generate a fluorescent signal, get some fluorescent light emitted from your sample, and then collect that using some kind of imaging device, everything that I'm about to talk about here and Jordan will talk about in the next section, all of that is going to apply no matter what type of technique you're using to generate fluorescence. Now, after I've gone through a lot of the introduction of what we mean by fluorescence, this is the image that tends to be built up in our heads, where we shine some color there at our sample and we get some different color glowing off of our sample. But in reality, it's actually a little bit more tricky than that. Typically, we have to flood the sample with our excitation light, and the sample actually emits a very small amount of the emission wavelength. And so we often have to employ the use of many different filters to cut out the flooding excitation light so that we can actually see the light that we're interested in that's being emitted by the sample. Now, to do this, practically speaking, There's usually at least two different filters. There's first an excitation filter that's used to filter white light coming from the light source into just the excitation wavelengths that we're interested in. And then there's a separate emission filter that's used to filter out the excitation light that's flooding the sample so that we just see the emitted light coming from the sample. Now, practically speaking, this is also employed in many different ways, but one of the more common techniques is a filter cube. So I just wanted to demonstrate this here, since many of the microscopes that you tend to work with these days will use a filter cube to do this. This filter cube contains both the excitation and the emission filter, but it also contains a special mirror that we call a dichroic mirror, which allows some wavelengths to pass while blocking other wavelengths. And since this has gotten to be a bit of a busy diagram, let's just walk through it step by step really quick. First, we have white light generated by the light source. It then passes through the excitation filter, and we end up with photons of our excitation wavelength. Those then bounce off of the dichroic mirror and are focused by the objective lens onto our sample, which the sample then absorbs. The sample then emits light in our emission wavelength. Those photons are collected and focused by the objective lens. And then this time the emitted light passes through the dichroic mirror. And then lastly, we have it passing through the emission filter just as a way of cleaning up any extra stray light that may have gotten around the dichroic mirror. And then finally, we capture this emitted light with our imaging device. Let's just look at how this actually works, practically speaking, with one particular molecule. We're going to look at the GFP spectra again. And with the excitation filter, it'll fit neatly around where the peak, where the bulk of the excitability of this particular protein is. And similarly with the emission filter, it's going to fit very cleanly over the emission peak, or the peak of the emission spectrum. And then the dichroic mirror will be sort of straddling the line in between, where we'll have a very low transmittance for the excitation wavelength so that it bounces off the mirror. And then we'll have a very high transmittance for the emission wavelength so that it passes through the mirror. All right, now this has already gotten pretty complicated, and I've just talked about one protein and one filter cube. And in reality, if you're using a lot of different fluorochromes, you're going to have a different filter cube for each one of them. And if you're not using filter cubes, you'll have to worry about filters and mirrors being moved around. But I just want to mention really quick that we actually make this pretty simple in our software. If you have motorized microscope components that can move all of these filters and mirrors around, we can actually control them with the simple click of a button to get all of those different pieces in the right place. So you don't even have to think about aligning all of these different components. Now, talking a little bit more about gathering the image data, I want to pause for a moment before we look at the actual acquisition process and talk about what we can do with the image data to help improve contrast after the fact. One of the most important tools for doing that is the image histogram. And before I talk a little bit more about how we can work with the image histogram, I want to just mention what data is being represented by it. So here I just have a sample image where it's a white background with a black oval drawn on the top. And the histogram x-axis is going to represent the brightness or the intensity of those pixels. And since right now we just have black and white, there's just two categories of bars for these histograms. Then the y-axis is showing the number of pixels in the image that correspond to that particular brightness or intensity. And so as we would expect, there are a lot more white pixels in this image than there are black pixels, because we have many more white pixels in the background and very few black pixels representing the oval. Now, in reality, it's going to be a lot more variation in your image than just simple black and white. You'll have many different shades of gray from black to white. And so your histogram is going to look something more like this. Now let's talk a little bit more about those tools I mentioned. We have the black point, we have the white point, and we have the gamma that we can use to adjust how the histogram is displayed on the screen as a way of getting better contrast on the parts of the image that we care the most about. Now, I'm going to go through these tools very briefly and just show what the effects are that they have. But what I want to focus on most is the steepness of the line that goes from the black point, or the black triangle on the left of the histogram, to the white point, or the white triangle on the right of the histogram. You can think of the steepness of this line as kind of equivalently being a contrast, or contrast over the part of the histogram that we're focusing on. So first off, let's look at the black point. The way that this works is that as we slide the black point from the left to the right and go towards brighter values of the histogram, we tend to lose contrast in the darkest parts of the image, but we increase the slope of that line for the brightest parts of the image. So we can get slightly better contrast in the brighter parts of the image by sacrificing the contrast in the darker parts. The white point kind of works the same way, but in reverse. As we slide the white point from the right to the left, going towards darker values, what we end up doing is sacrificing contrast in the brighter parts of the image to gain contrasts in the darker parts of the image. These tools can actually be used together to form what we like to think of as a window, where we're centering in on just a specific part of the histogram that represents the part of the image that we're interested in investigating. And so you can see here, I can adjust this window to focus on different parts of the image. The other thing that we can do is we can adjust the gamma, which changes the linearity of this line. But again, I want you to focus on particularly the slope of this line. On the top image, you can see that the slope of the line becomes flatter and shallower as we go towards the brighter parts of the image, but it becomes steeper towards the darker parts of the image. So we're sacrificing contrast in the brighter parts to get contrast in the darker parts. And at the bottom, we have a flatter line near the darker parts of the image and a steeper line near the brighter parts of the image. And so we're kind of doing the reverse, where we're sacrificing contrast in the darker parts to gain contrast in the brighter parts. And what I want to point out is that these settings can all be used in tandem, and different parts of even the same image might have the best contrast possible by using varying different settings with these. But the other thing that I want to make clear here is that, as you can see, I'm able to get better contrast as I work with the brightest parts of the image near the bottom. Near the top, I'm looking at a very dim signal. And so even though I can increase the contrast and start to see what's going on with the dimmest parts of the image, ultimately, the better that you can do to get a brighter signal to really fill out that histogram window at the time of acquisition, the more data there is for us to work with the histogram later. So now that I've gone through a couple of the tools and sort of the background of fluorescence, I just want to demonstrate what acquiring a multi-channel image looks like in our software. So first, the sample that I'm going to be using here is going to have 3 different colors. We're using DS Red, GFP, and BFP. I'm going to start with looking at the DS Red channel, and it's pretty dim. I'm going to start increasing the exposure time, but I've quickly gotten to about 1,000 milliseconds of exposure time, which is pretty high for most cameras. So to compensate, I'm going to try and move the white point on the histogram until I can see more of the image in the red channel. Then I'll switch over to the green channel, and I can already see it pretty well, but I can definitely increase the exposure time to get better brightness, to get better contrast on the brightest parts of the image. Now, and oh wow, the blue histogram is way too bright. So I'm going to use the clip detect tool to highlight all of the areas in red in the image that are overexposed. And all I have to do is reduce the exposure time on the brightest red parts of the image until I don't see any more of those red pixels. Then I can turn off the clip detect tool and I can hit the acquire button, where the software is going to then drive through all of the different filter cube positions and give me a multi-channel image. Now that I've acquired the image, I can do a little bit more to clean up how these channels look. I'm going to start again with the red channel here. I can move the black point in to try and remove some of that background that is kind of hanging like a red haze over the image. Once I've done that, I'll move on to the green channel, and I'll do the same thing. There's far less background in the green channel, but I can still move the white point in a little bit to get some brightness. I can still move the black point in to remove some of that background and get rid of some of the green haze. Now, when I'm looking at the blue channel, we're going to repeat the same process of moving the black point and the white point to get better contrast. But I find it very difficult to see when it's a pure blue on the screen. So I'll just quickly change that coloring to white instead. And it becomes immediately a lot clearer what needs cleaning up, and the fact that I can actually get a little bit more of that background removed by moving the black point. This actually brings up a very interesting point, which is that whenever we acquire a multi-channel image, it's always going to be acquired as a series of monochrome or black and white or grayscale images. The color that you see is a false color that's put on top of these channels as a way for us to visualize the data a little bit better and make more sense in our heads as to what's going on in the image. But using the white to go back to the pure monochrome can often be really helpful in figuring out how much background you have, whether you can increase the contrast, and just in general figure out what the best settings are for each channel individually to make the information in that image as clear as possible. So for instance, when I go back to the red channel here and I flip it back to white, it becomes immediately apparent that I can do a lot better in terms of contrast. The background is still pretty bright. I can reduce that a bit. Oh, that looks a lot better. I'm definitely getting more of the actual stained cells showing up in the channel now. So I'll make a couple more adjustments just to see if I can get the contrast to be as good as possible, remove as much of that background as I can. And then that's starting to look good. So I'll return the channel back to a reddish color. I've changed the blue channel to a lighter blue, more of a cyan color, just to make it a little bit easier to visualize. And now I can turn all of the channels back on. And this is the final image that we have. A lot better contrast than the original image as it was acquired. So now that we've talked a little bit about the acquisition, let's just go through some common issues that tend to come up a lot. The very first one that we see by far the most common is that the image itself is completely black or appears to be completely black. This could be just simply that the light path hasn't been set up properly, that the filters and the mirrors aren't quite in the right positions. And as I mentioned, we make this pretty simple in the software, just clicking a button to rotate all of those different pieces. It's also possible that the camera exposure time is too low. You saw with the red channel, I struggled for a moment there to get a good enough exposure time that I could start to see the image. And it could also be that the histogram needs adjustment, similar again, like what I did with the red channel and the white point. We also need to consider the fact that it might not be any of those different imaging settings. It might just be that there's not enough light getting to the sample, so you need to turn up the light source a little bit. Now, to go back to this image, on the left, you can see the histogram that I have is actually very thin and small. But once I've gone to the right image, where I've increased the exposure time, I'm now taking advantage of much more of that window, that histogram window. And this actually brings up a very good point that we often talk about a lot when we're doing digital imaging. These two terms, dynamic range and bit depth, if you speak to us a little bit more about the imaging part specifically, you'll probably hear us use these terms quite a bit. And I just want to mention what they represent. Dynamic range you can think of as being sort of the biggest difference that your camera can see within a single image between very dark signals and very bright signals. And so the more dim signals it can see at the same time as very bright signals, the higher dynamic range we have. You can think of that dynamic range of your camera as being represented by the actual histogram window itself. The bit depth is a little bit more tricky to understand, but I like to sort of think of it as the number of steps that you get between the darkest and the brightest value, or the number of intermediate gray values that you get between black and white. The higher the bit depth, the more close you get towards a true continuous curve from black to white. But there are many reasons why you might reduce that bit depth, and that's something that you can talk to us a little bit more about if you're interested in your particular study. So just to demonstrate how dynamic range comes into play, let's look at another example where the image appears to be grainy or pixelated. In this case, it's actually a combination, most likely, of histogram settings needing adjustment and the camera exposure time needing adjustment. And so to go back to this image, I've both increased the exposure time and I've moved the white point out to get a bigger window in that histogram. And the difference here is that I'm really taking full advantage of the entire dynamic range of the camera in the rightmost image, where I've increased these settings. And you can see that the grainy appearance tends to disappear because we're using much more of those intermediate gray values that the camera has. And so we're getting better detail. And last but not least, it's also possible that we can see that the actual sample is present, but we're just not able to see where the fluorochrome has stained the sample. Typically speaking, we would want to start with all of the other things that I mentioned. Look between mismatches between the filters, look at the histogram or the exposure time, see if those need adjustment. But we also need to consider that the background might just be too high, or the fluorochrome itself might be too weak. Or even in the most ultimate of circumstances, it's possible that the fluorochrome at that particular part of the sample just isn't present. And so you're just not seeing any of that emitted signal. Now, in this particular case, I am able to make some histogram adjustments without changing the exposure time to get better contrast, to remove some of that background and to get better staining on the cells to show up. But the other thing that I want to point out here is that there is a limit to what we can do with the histogram. We can use that data that we've already acquired and we can display it a little bit differently to make it more interesting and more obvious what's going on in the image. But the better that you can do with getting a brighter signal to start with as you're acquiring the image, the more data we have to work with and the more effect your image histogram adjustments are going to have on the final image. So I kind of breezed through a lot of really big topics really fast. But if you want to learn more about any of these in depth, I can recommend a few different resources for you. The first is Microscopy U, which goes through a lot of introductions to many of the things that we're talking about here. And it also takes some deep dives into the microscope hardware itself and what its capabilities are. Next, we have Chroma's SpectraViewer. Chroma is a company that makes filters and filter cubes and dichroic mirrors, and we tend to use a lot of them with our systems. And they have a fantastic tool called the SpectraViewer that allows you to actually dive in and see how the different spectra of excitation and emission are going to interact between your filters and your fluorophores. Next, we have FPBase, which is a fluorescent protein database. This will give you a lot of information about both the fluorescent behavior of the proteins that we're talking about and also the chemistry that's involved with them. And then last, we have Dr. Dan Peterson's NeuroRenew workshops. These workshops are intensive introductory courses that get you into how to actually use the devices, use the microscopes, as well as a lot of the theory behind the imaging. And Dan Peterson's courses have been absolutely instrumental in training up our support staff here. So I would highly recommend, especially for new beginners, that you check out some of his materials.
Awesome. Thank you so much, John. That was fantastic. Great background, great base of knowledge for everybody. Jordan, can you take things a little further with the slide scanning?
Yeah, so we're going to extend off that. And like Nate said, we're going to talk about how to translate this into slide scanning. So slide scanning is an additional module that is offered for Neurolucida, Stereo Investigator, and Microlucida, and it's utilized to create high resolution 2D images or 3D stacks at any magnification. This can present a ton of benefits, such as you can use SlideScan to create a virtual record of your tissue that can be offloaded for continuous future use. And this allows users to continue analysis offline away from the microscope system, which can be a great benefit for core facilities to have more available time on that scope system. This also extends the research lifetime that comes from your sample by having a permanent high-resolution virtual record past the time when your physical tissue could have bleached and become unusable. Lastly, slide scanning can be used with a wide variety of research methods, including bright field microscopy, wide field fluorescence, as well as structured illumination and confocal devices. And today I'm going to highlight how to use slide scanning with wide field fluorescence. Before I dig into some demos, I'd like to highlight how we can elevate the fluorescent slide scans by incorporating the Vesalius. This is an additional component NBF has recently added to our product line, and it's a cost-effective spinning disc confocal microscope attachment that is great for quick, large-scale 2D image and 3D stack acquisitions. The Vesalius can also be taken advantage of with our slide scanning workflows to create crystal clear slide scans for your lab. It's an easy add-on to create huge improvements in imaging results, and these sample comparison photos show what is possible with the Vesalius with the sharp improvements in reducing out-of-focus light and creating a high signal-to-noise ratio. And before moving on to the demos, let's highlight some key differences between taking a two-dimensional and three-dimensional slide scan. So to start, a 2D slide scan can create a 2D high-resolution image at a single plane of the sample. And to do this effectively, users need to consistently define a uniform focal plane and prevent creating a final image with varying focal levels in the sample. While in comparison, a 3D slide scan creates a high-resolution three-dimensional stack of the sample, and to do this effectively, users need to consistently define focal heights to be at the top of their sample. A good tip is to focus just above your tissue and set the height for each focus site just when your tissue starts to come into focus. So I'm going to move on to doing a demonstration in 2D of a 2D slide scan in Stereo Investigator. So to start, we're going to open up our workflow, and this can be done by going to the Acquire tab and selecting Slide Scanning Workflow. This workflow is going to help guide us through all of the necessary steps to take a successful slide scan. So in the first setup step, we need to define the base settings of our scan. And so for this demo, I'm going to set the Acquire type to be a 2D slide scan and have my illumination type set to fluorescent. I can also take a single channel or multi-channel fluorescent slide scan. And today I'm going to choose to demonstrate taking multi-channel scans, so I'm going to leave that box checked. The scan type setting allows us to decide how we're going to identify our region of interest. So I'm going to choose to scan inside of a contour only, which is going to allow me to trace a contour later in the workflow to define my region of interest. Lastly, we need to set our export locations for our data and image files produced from the scan. And we advise setting these to be local destinations on your computer. And if you'd like, you can always upload them to any online or network storage afterwards. And in the scan settings step, we can define the stage settle delay and overlap trim. The overlap trim is an important aspect that defines the overlap between each tile image during the slide scan. Additionally, this overlap is used when stitching is enabled to adjust for minor discrepancies that can be caused by thermal drift, stage movement inaccuracies, and other factors. While the stage delay setting causes the algorithm to wait every time the stage moves to a new tile, and this can help reduce any blur you might see in your final image. As we move on to this third step, I'm going to set the magnification I'm going to use to trace the contour. So I'm going to choose to use 5x, and then I'll move on to the trace section. And this is going to allow me to trace my contour around my region of interest. I'm not being too particular with it. I'm just looking to set the bounds for the algorithm to trace what I would like imaged. And the next step, I'm going to set my Acquire magnification. This is the magnification the slide is going to be imaged at. So I'm going to choose to use 40x to create that high resolution image. And I'm also going to use this time to help set up my multi-channel acquisition settings like Jean presented earlier. And so similarly, I'm going to be using the DS-Red, GFP, and BFP channels. So I'm adjusting the black and white points and getting a good exposure for each channel before moving on to just kind of set my base point for my scan. And as I move on, you might notice that the workflow skipped the step for region selection, and that happened because I am scanning inside of a contour, and the region selection step isn't necessary as I only set one available region for it to scan. And as we move on to the background correction step, I'm not going to utilize background correction here, though it can be a very useful tool to minimize the effect of uneven illumination and tiling effects you might see in your final result. Similarly, for taking a sample acquire, this is an optional tool that can be used to take a smaller test slide scan with your current acquisition settings. It can be very beneficial for labs taking long slide scans that can range for a few hours or even days. It's a great double check to ensure you're going to get a quality final result before investing that time into the system. And this focus map step is a critical component of the slide scanning workflow. By indicating a uniform Z position across a few focus sites, the algorithm will create a focus map that is used to set the Z height for every tile in the image. So to start, we can manually define our focus map by selecting a few focus sites throughout our sample. And after manually selecting a few sites, I'm going to hit Focus at Selected Sites. From here, it's important to consistently focus on similar focal planes to create a uniform looking image. And so setting an inconsistent focal plane between tiles can create a very inconsistent final product as your tiles are going to be imaged at varying focal heights throughout the slide scan. A good tip that I like to use is use the Z meter on the right to just kind of eyeball my consistency and just make sure I'm being uniform throughout my focus map. And after completing this focus map, you can also enable the Show Heat Map setting to see how uniform the focus map is. If you have some sites marked as red, this indicates the value is seen as a large jump in Z position from adjacent tiles, and it's a good spot to double check your focus map before starting the scan. And now we're going to continue on to the final step, where we now have some final options we can check before initiating the scan. And this is where we can also enable stitching to use the tile overlap we had defined earlier to better align our tiles. And we can also use this slider to change the compression ratio for the final image. And another useful feature is observing some elements in the scan summary. This displays your defined scan settings, as well as previews some final information like expected storage size of your scan. And when I'm ready, I can hit start scanning with the focus map to initiate my slide scan. So as we move on here, I cut out the time spent scanning the sample. As a note, this can be a varying length of time depending on how many tiles you're scanning and how many channels you're using. But when it's all done, we'll get our final compiled image showing all of the tiles imaged together. Now I'm going to move on and do a demonstration of a three-dimensional slide scan to just highlight some key differences between the two. So for our 3D scan, we're going to start by reopening the slide scanning workflow again. And now in my setup step, I'm going to reset up my settings for a different three-dimensional scan. So for my acquire type, I'm going to set it to be a 3D slide scan and still have my illumination type set to fluorescent. Similarly, I'm going to keep multi-channel enabled to allow myself to take a multi-channel three-dimensional slide scan. However, this time I'm going to set my scan type to be a 3x3 grid. And additionally, we will need to set our data and image file locations still. Remember, we still want to save these locally, and we can always upload them to any online or network storage after the scan is complete. And as we move on to this scan setting step, we now additionally need to identify our stack options. So this requires you to have some background knowledge of your specimen before initiating the scan. You'll need to know the height of your tissue and your desired stack parameters. So in my situation, I'm going to choose to set my desired height to 40 microns and the distance between each plane to be 4 microns. The result of this will be the software taking a 40 micron thick stack at 4 micron intervals at each tile in my 3x3 slide scan. I'm also going to leave my overlap at 11%, and as a note, this needs to be greater than 10% if you wish to enable stitching later in the slide scanning workflow. And as I move on from this step, you're going to notice that the tracing magnification and trace steps are bypassed here, as I'm not tracing a contour from my scan. Instead, it's going to be that 3x3 grid I defined earlier in the workflow. However, I'm still going to need to set my acquire magnification for what the slide is imaged at, and I'm going to use 40x again. I'm also not going to utilize background correction or sample acquire for this 3D scan. And as I mentioned last time, background correction can be used to assist with any tiling or uneven illumination being seen in your final result, while the sample acquire step is used to take a small test slide scan to test your set parameters, and it's a great tool to utilize for labs taking large scans lasting a few hours or even days to get a preview before starting their scan. And now as I move on, I'll need to define my focus map for my 3D slide scan. And so in this focus map, you can see a marker I had previously set before starting this workflow. And this can be utilized to remember to return to this marker later as a focus site. And so by hitting the button, use markers as focus sites, the software is going to automatically put a focus site anywhere it finds a marker. Afterwards, I can also manually add a few more here to just make a more well-rounded focus map. And so now a key difference between taking a two-dimensional and 3D slide scan is that for a 3D focus sites, we want our focus for each site to be at the top of the tissue. Whilst for 2D scans, we will uniformly focus across our desired focal plane. Essentially, for the 3D scan, we're going to be setting the top of our tissue with our focus map, and the algorithm is going to take a stack downwards at each tile to compile our final image. If you watch me go through my different focus sites, I'm actually using the Z meter on the right as a useful reference to just get to the top of my tissue, to just know I'm above out of focus, and then I can hit my focus map just as I start to see stuff come into focus. And I'll need to consistently repeat this process for every focus site. And as I finish up this focus map, remember you can also use the heat map as a double check to validate your created focus map. And as we move on here, before starting our scan, we can also review this final step for our key information. So since 3D slide scans can be much larger, I find this to be a good time to double check the estimated required storage size for the scan. And additionally, you can also enable stitching to help get that crisp aligned final result in your image. And as we get started, just as another note, 3D slide scans can take much larger than 2D scans as the system is just taking many more images. So in this example, the algorithm is taking nearly 100 images, with 11 being taken at each of the 9 tiles, whereas if this were a 2D 3x3 slide scan, the software would have only needed to take 9 images total. And after the scan is complete and compiled, I'm going to start by reopening the produced image, and from here, we can go through the individual planes in the stack that was obtainable from the 3D slide scan. We can also go through some post-image adjustments that Jean had demonstrated before. And so as you can see here, I can navigate to the green channel and adjust my white point to just see if I want to get a bit more signal, a bit better visual out of that channel. And as I conclude these demos, I wanted to cover a few mistakes that could cause some unexpected slide scanning results. So to start, in this image, you can see 6 arrows that match 6 tiles that were included in this slide scan. These arrows identify a black silhouette that is seen throughout the image and was imaged at each individual tile. This was caused due to an aperture being left in the light path, blocking off some light from reaching the sample. Troubleshooting this light path that Jean went over is a great skill to have for effective fluorescent imaging and slide scanning. And if you ever need any help, please reach out to our support team for assistance. And now in this example, we have a few things going wrong with this. So to start, you can notice a rather large unalignment from the middle tiles of the slide scan. This was due to poor stage and camera alignment, and this alignment is a critical component of the slide scanning workflow. So this means that the camera needs to be perfectly aligned with the stage so that the camera visually matches what is happening with the stage movements. And so in this example, the camera was very crooked, creating that large disconnect and unalignments you can see between each tile in the scan. And this can range in effect, with some being very apparent, like in this image, or being extremely small and only found when zooming in between the tiles of your slide scan. Our user guides for our products have a great guide to help assist with this alignment process, and additionally, enabling stitching during the final step of the workflow can also help improve any of these slight misalignments you could see, and these can stem from other factors such as thermal drift or stage movement inaccuracies as well. However, another component going on in this image is the tiling effect, showing some uneven illumination between each tile. This uneven illumination can be caused from a varying number of factors, and we offer a few solutions to work with this. So some options could be to try using background correction, ensuring that your fluorescent light source is centered and aligned. A good tip is just to reach out to our support team if you would like any assistance with this, and we can help troubleshoot with you based off of your specific microscope setup. And so Nate, I'm going to pass it back to you here to help guide us through our Q&A period.
Awesome. Thank you so much, Jordan. That was fantastic. So let's take a look at some of the questions we have in here. Let's see. All right. So we have a question. Is the software capable of scanning multiple regions of interest at the same time?
Yeah, so in my demos, in both of my 2D and 3-dimensional demo, it actually skipped over that step. And that was just because you need a few parameters to be true for that to happen. So if you're scanning a grid, that's not going to happen because you're just going to be scanning the grid and not the contours. But if you were to set the contour like I did in the 2-dimensional scan, you can actually trace multiple contours, and the slide scan can trace both of the contours during the workflow process. And this can be great for having multiple slides or multiple regions of interest. So say if you have a custom slide holder, you can have multiple slides being imaged during one run of the workflow. And it's a great tool to just help expedite some of this imaging process.
So yeah, we can do multiple sections as well. So you have multiple sections on the same slide, or as Jordan said, a slide holder that can accommodate multiple sections, multiple slides rather. We can do that as well. We're really limited by the size of the stage and the slide holder. And your disk space. We have customers that are running scans for days, running, 10 or 20 sections that they're acquiring. So it's only limited by the physical piece there. So the software can kind of handle whatever you can throw at it. All right, let's look at the next question we have here. Okay, so I'm doing scans, but I can see tiles and seams in my final image. How can I fix that?
Yeah, so that's a very common thing. A good recommendation, like I said, is just to reach out to our support team, and we can help notice something that could be going wrong with your specific hardware setup just based off things we've seen before. And another useful tool is the background step that you can utilize to help mitigate some of this tiling, where the software can actually just help make that clear image and make it a bit crisper for you.
Okay. What are the proper imaging settings, e.g. gain, exposure time, etc., for very weak fluorescent signal samples?
So as you saw with the sample that I had, actually one of the channels was pretty weak. The red channel wasn't giving me nearly as much signal as the rest of the image. So it really depends on how you can fill up that histogram, the adjustments that you can actually make there. As we mentioned, you can change the exposure time, you can adjust the histogram settings, you can work with increasing the intensity of the light source. Realistically, there isn't necessarily a one-size-fits-all answer for this. It really is highly dependent upon how your sample looks and the different protocols that you are working with, as well as the image device that you have. But realistically, if you're able to get at least some kind of signal, there's a lot of different tools in the software that we can use to make the most out of the signal that you do have.
Nice. This one kind of follows on that question. Are there any recommended limits for exposure, dynamic range, and light intensity?
Mm-hmm. So typically speaking, this is also going to be one of those sample dependent things, but a good rule of thumb, at least in terms of exposure time, is that if you start going above about 1,000 milliseconds of exposure time, things tend to be so delayed in between each frame that the camera can acquire that you might end up accidentally exposing your tissue too much and causing a lot more photo bleaching. Whereas if you go a little bit below 100 milliseconds, typically most monochrome cameras that are designed for low light signals like you would have in fluorescence, they don't really like to go much further down below 100 milliseconds, so you can start to see some interesting behavior there. But other than that, there really, again, isn't necessarily a one-size-fits-all here. It really does have a lot to do with just how your sample has been prepared and what's available.
Yeah, I think one of the things that I've tried to do is keep the exposure, like when you start to get above a second for your exposure time, it's really going to impact both what you see on the live image. There's going to be a lot of lag and delay. Sometimes we get into that and people are like, oh, you know, my image is lagging. And really, that's just a product of the exposure time. When you have a 2-second exposure time, that's really like when you think of, you know, a film camera, right? Like obviously we're doing CCDs here, but a film camera, that exposure time is how long your shutter's open and exposing the film. Here, you know, it's how long we're gathering light in here. So when you have a 2 or 3 second exposure time, you know, we've got to wait for that 3 seconds to expose the chip and gather light. And so you're really going to start to have some long acquires. So if you're doing one of those big scans like Jordan was showing us, your scan is really going to start to get quite long if you have 2 and 3 second exposure times. It's going to add up a lot. So when you're doing hundreds or thousands of images, you got your image stacks going, you know, it really adds up very quickly.
And I think as a note, just like remember what Jean said, it's just remember the consequences that come with it, where you're going to have to expose your tissue to more light, and it can result in some of the bleaching and other effects just stemming from that.
And the one last piece I did want to mention about that, because part of the question we had there was about intensity. There was a question about dynamic range too. One thing about that is bigger is better with dynamic range. The longer you can get that within reason, like obviously we talked about exposure. We don't want you to have a 10-second exposure time so you can get that wide dynamic range. But within reason, try and get that as wide as you can, because, you know, you can have all the image processing in the world afterwards, but the better quality image you can get at acquire time, things are just going to be better for you. You get that really narrow dynamic range, it's a little harder to work with. As Jean showed, there's a lot of really good tools for that, but you want to try to get that as wide as you can. And then part of that question was the intensity. A really great tool for that is the clip detect feature. What you want to avoid is any sort of clipping. So you don't want it to be too bright, so you're losing signal, or too dark, so you're losing signal. And you can see on that live histogram if things are beyond your black and white point. So that's really the guideline there. So use that clip detect feature, and you'll see the pixels will show up as clipped, red and blue, red for the bright and blue for the too dark. And you can see what kind of information's getting lost, basically. So use that clip detect. That will help you with your intensity. All right, how are we doing here? All right, so we have a question. How big can the depth of field be? This particular attendee was scanning 40 micron sections. Can we cover the entire thickness there?
So this is going to be fairly dependent on the hardware that you have, but at least in terms of what hardware you do have, our software is going to be able to handle whatever the thickness is that you've got there within the working distance of the objective that you're using. So it's important to consider that, well, two factors really. With your objectives, each objective is going to have a different depth of field over which in Z, things will all appear to be in focus, and typically a thinner depth of field from a higher magnification objective is going to mean a better Z resolution. And so you'll be able to get better detail as you go through the different Z planes. But perhaps the other thing that's going to be more important to worry about here, especially as you start getting to thicker and thicker samples, is that each objective has a different working distance, or the maximum distance that it can actually see into the tissue before you end up with the situation where the objective makes contact with your cover slip, which is not something we don't want to go there. We can end up breaking slides if we get too close. So we need to take into account both the depth of field of the objective itself for the purposes of Z resolution. We also need to take into account the working distance of the objective and how thick the sample is in relation to it. But really, again, because this is going to be so dependent on the hardware that you're using, if you have any specific questions about this, definitely email the tech services team and let us know. We can talk and discuss this with you.
Another piece of that as well is that there are longer working distance objectives, so that's something that we can help with, and you can talk to our team when you're figuring out your system. Okay, so another question we have is, how does the system handle different intensities for different labels or different channels?
So one of the things that some of you may have noticed as I was going through my demonstration and adjusting the different settings for the three different channels was a little remember video settings checkbox underneath all of the buttons for the controls for the different channels. That's really the magic button here in our software. That's what allows us to make all of these individual changes from one channel to another so that each one is able to be acquired with the best settings to get the best contrast. My sample, as you saw, the red channel was very weak, the blue channel was very strong, the green channel was somewhere in the middle. Basically, as long as you can see with relative contrast the different channels that you're working with, and make sure that little checkbox is switched on, we can handle any sort of intensities that you have on your sample, again, sort of limited by the dynamic range of your camera. And as Nate mentioned earlier, when we're talking about differences in intensities and varying signal levels, bigger is better for dynamic range. The bigger of a dynamic range your camera has, the more differences in intensity you'll be able to acquire on the same sample.
Perfect. All right, so it looks like we're coming up on our time here. If we didn't have a chance to get to your question, we have those, and we can get back to you with those answers. Thank you again, everyone, for joining us today. We really appreciate your time. Again, thank you for the questions. That really helps the discussion. It helps us, it helps the presentation, it helps everybody else, because if you have that question, odds are somebody else probably does as well. So if we didn't get to your question, like I said, we'll try to get back to you. But also, please feel free to email us at support@mbfbioscience.com. We're here to help you. That's what we're here for. You're not bothering us. We want to help you. We want to help you be successful, use our systems. You know, so whatever the question is, let us know and we'll try to help you out. Additionally, we have a forum as well, so you can look for an issue that you may be having. You can post questions or just discuss topics that you're running into and that you're having. This webinar has been recorded. We'll get that up on the website in the next couple of days. You know, you can refer back to it, or if you have a colleague that couldn't join today, you can forward that to them. Lastly, we do have a survey after the webinar. Please take a few moments to fill that out. We love hearing from you, you know, what you liked, what could be different, maybe even other topics that you'd like to see in a future webinar. So thank you for doing that ahead of time. And again, thank you so much for being with us today. If you need anything, we're here. Hope everybody has a great summer and take care.